Hello everyone, my name is Nur Shafizawani. Let's start with electron carrier molecule.
Electron carrier molecules are essential for life as we know it. They shuttle electrons, the currency of cellular energy, through complex pathways that ultimately power everything from muscle contraction to the synthesis of the
NAD+, FAD, and ADP+, are the examples of electron carriers.
NAD+, or nicotinamide adenine dinucleotide, is a biological molecule that plays an important role in cellular energy transfer. In the reaction, NAD+ is reduced to NADH, which can then be used to store energy within the cell. The reactants, NAD+ and two hydrogens, are shown at a higher energy level than the products, NADH and a hydrogen ion. This indicates that the reaction is exergonic, meaning that it releases energy. FAD, or flavin adenine dinucleotide, plays a crucial role as a redox cofactor in numerous metabolic reactions, particularly in energy production. FAD is involved in both catabolic, breaking down molecules for energy, and anabolic, building molecules pathway. FAD can accept two hydrogen atoms along with their electrons. This process leads to the reductions of FAD to FADH2. Here, FAD gains two hydrogen atoms, each carrying one proton and one electron, resulting in the formation of FADH2. NADP, or nicotinamide adenine dinucleotide phosphate, plays a crucial role in the light-dependent reactions of photosynthesis. It accepts electrons from photosystem 1 during the light reaction and is reduced to NADPH, which is used to convert carbon dioxide into carbohydrates during the Calvin cycle. NADP accepts electrons and protons from donor molecules in redox reaction. The reduction of NADP to NADPH involves the addition of two electrons and one proton, resulting in the formation of NADPH.
Glycolysis is a metabolic series of reactions that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvate. Moving on to the functions of glycolysis. The first function is energy production. Glycolysis produces a net gain of 2 ATP molecules per glucose through substrate-level phosphorylation. The second function is production of NADH. Glycolysis generates 2 NADH molecules per glucose, molecules which can be used in oxidative phosphorylation to produce additional ATP under aerobic conditions. The third function is biosynthetic precursors. Intermediate of glycolysis, such as glucose 6-phosphate, fructose 6-phosphate, and 3-phosphoglycerate, serve as precursors for the synthesis of nucleotide. The last function is hormonal regulation. Glycolysis is regulated by hormones such as insulin and glucagon, which influence enzyme activity and the availability of glucose. Glycolysis is a series of 10 enzymatic reactions that convert glucose into pyruvate, generating small amounts of ATP and NADH in the process. Here's a step-by-step breakdown. Starting from phase 1, where energy investment involves 5 steps, where the first step is to undergo hexokinase reaction, where glucose is phosphorylated to trap it in the cell. Secondly, phosphoglucose isomerase reaction, where glucose 6-phosphate is rearranged into fructose 6-phosphate. Thirdly, phosphofructokinase reaction, where fructose 6-phosphate is phosphorylated to fructose 1,6-biphosphate. Fourthly, aldolase reaction, where fructose 1,6-biphosphate is split into two 3-carbon sugar. And the last step for phase 1, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate through triose phosphate isomerase reaction. The next phase is phase 2, which is energy payoff. Firstly, involve glyceraldehyde 3-phosphate dehydrogenase reaction, where it oxidizes and phosphorylated producing NADH. Secondly, phosphoglycerate kinase reaction, where 1,3-biphosphoglycerate transfers a phosphate to ADP, forming ATP. Thirdly, 3-phosphoglycerate is rearranged to phosphoglycerate through phosphoglycerate mutase reaction. Fourthly, enolase reaction involve two phosphoglycerate is dehydrated to form phosphoenol pyruvate. Lastly, pyruvate kinase reaction where phosphoenol pyruvate transfers a phosphate to ADP, forming ATP and pyruvate.
Hi, my name is Noria Shodira. So I will continue with transition reaction, which is happen after glycolysis and before entering the Krebs cycle, or we call it as citric acid cycle. So transition reaction take place in mitochondria. So its purpose to prepare two carbon unit of acetyl coenzyme A for entry into the citric acid cycle, which is Krebs cycle, where further energy extraction through oxidation occurs. So for the outcome, the transition reaction will produce one molecule of acetyl coenzyme A per molecule of pyruvate, and it will enter the citric acid cycle, Krebs cycle, to generate more ATP through oxidative phosphorylation. So I will continue with the process. So basically, in the transition reaction, pyruvate will convert into acetyl coenzyme A. There's an enzyme involved in this transition reaction, which is it is catalyzed by the pyruvate dehydrogenase complex (PDC), which consists of multiple enzymes and cofactors. And it is start with pyruvate. Pyruvate consists of 3 carbon units, and when it is oxidized, it releases carbon dioxide and form an ADH. Then the remaining 2 carbon unit, it will bind with coenzyme A and form acetyl coenzyme A. Okay. In summary, pyruvate is the product of the glycolysis will convert into acetyl coenzyme through transition reaction in mitochondria, and it releases carbon dioxide and NADH in the process. Then the stage this sets the stage for further energy production in the citric acid cycle, with each it will be ready for the next stage, which is Krebs cycle.
Today, I'm going to explain the Kreb cycle, also known as the citric acid cycle or the carbolic acid cycle. This cycle is crucial for energy production in aerobic organisms and occur in the mitochondria. The first step of the Kreb cycle is the formation of citrate, where acetyl coenzyme A combines with oxaloacetate to form citrate, catalyzed by the enzyme citrate synthase. Next, citrate is rearranged to form isocitrate through a reversible transformation facilitated by the enzyme isonicitase. In the third step, isocitrate is oxidized to produce alpha-ketoglutarate, catalyzed by isocitrate dehydrogenase, resulting in the reduction of nicotinamide adenine dinucleotide NAD+ to NADH and the release of carbon dioxide molecule, marking the first decarboxylation step. Following this, alpha-ketoglutarate undergoes another decarboxylation facilitated by alpha-ketoglutarate dehydrogenase, producing succinyl-CoA, NADH, and releasing another carbon dioxide molecule. In the fifth step, succinyl-CoA is converted to succinate, catalyzed by succinyl-CoA synthetase, producing geonosine 3-phosphate GTP, which can readily converted to ATP and coenzyme A. Succinate is then oxidized to form fumarate in the sixth step, with the enzyme succinate dehydrogenase reducing flavin adenine dinucleotide FAD to FADH2. In the seventh step, fumarate is hydrated to form malate, catalyzed by the enzyme fumarase. In the final step, malate is oxidized to generate oxaloacetate with malate dehydrogenase producing NADH. The oxalate can then combine with another acetyl-CoA molecule, continuing the cycle. The Krebs cycle occurs in the mitochondrial matrix, and its main purpose is to generate high energy molecules like NADH, FADH2, and GTP or ATP. For each acetyl-CoA molecule, the cycle produces 3 NADH, 1 FADH2, and 1 GTP, along with 2 carbon dioxide molecules as byproduct. Understanding the Kreb cycle is crucial for grasping how cells generate energy. This cycle is integral to cellular respiration and the production of ATP, which is vital for all cellular activities.
Assalamualaikum and hi, my name is Sharifah Ifai Marquis and I will continue this presentation with electron transport chain. Electron transport chain is a series of reactions that resemble a relay race or bucket bracket in that electrons are passed rapidly from one component to the next, to the end point of the chain where the electron reduce molecular oxygen, producing water. There are 4 complexes in electron transport chain, which is complex 1, Q and complex 2, complex 3, and complex 4. To start, 2 electrons are carried to the first complex aboard an ADH. This complex is composed of flavate mononucleotide FNN and an iron sulfur containing protein. FNN, which is derived from vitamin B, is one of the several prosthetic groups or cofactors in the electron transport chain. A prosthetic group is a non-protein molecule required for the activity of a protein. Prosthetic groups are organic or inorganic, non-peptide molecules bound to a protein that facilitate its function. Prosthetic groups include coenzymes, which are the prosthetic group enzyme. The enzyme in complex 1 is an ADH dehydrogenase and is a very large protein that containing 45 amino acid chains. Complex 1 can pump 4 ions across the membrane from the matrix into the intermembrane space. And it is in this way that the hydrogen-ion gradient is established and maintained between the 2 compartments separated by the inner mitochondrial membrane. Complex 2 directly receives FADH2, which does not pass through complex 1. The compound connecting the first and second complex to the third is ubiquinone, or we call it as Q. The Q molecule is lipid-soluble and freely moves through the hydrophobic core of the membrane. Once it is reduced, QH2 delivers its electrons to the next complex in the electron transport chain. Q receives the electron derived from NADH from complex 1 and the electron derived from FADH2 from complex 2, including succinate dehydrogenase. This enzyme and FADH2 form a small complex that delivers electrons directly to the electron transport chain, bypassing the first complex. Since this electron bypass and thus do not energize the proton pump in the first complex, liver ATP molecules are made from the FADH2 electrons. The number of ATP molecules ultimately obtained is directly proportional to the number of proton pumps across the inner mitochondrial membrane. The third complex is composed of cytochrome B, another iron sulfur protein, recenter and cytochrome C protein. Cytochrome proteins have a prosthetic group of heme. The heme molecule is similar to the heme in the hemoglobin, but it carries electron, not oxygen. As a result, the iron and its core is reduced and oxidized as it passes the electron, fluctuating between different oxidation states. The heme molecules in the cytochrome have slightly different characteristics due to the effect of the different proteins binding them, giving slightly different characteristics to each complex. Complex 3 pumps proton through the membrane and passes its electrons to cytochrome C for transport to the fourth complex of protein and oxygen. Complex 3 pumps proton through the membrane and pass its electron to cytochrome C for transport to the fourth complex of protein and enzyme. The fourth complex is composed of cytochrome protein C, A, and A3. This complex contains 2 heme groups and 3 copper ions. The cytochromes hold an oxygen molecule very tightly between the iron and copper ions until the oxygen is completely reduced. The reduced oxygen then picks up 2 hydrogen ions from the surrounding medium to make water. The removal of hydrogen ions from the system contributes to the ion gradient used in the process of the chemiosmosis.